Introduction. Professor. Good morning, class. Today, we will dive into the fascinating world of DNA methylation and its role in cancer. DNA methylation is an epigenetic modification that plays a crucial role in the regulation of gene expression. In recent years, extensive research has revealed significant associations between DNA methylation and various aspects of cancer, including genome instability, metastasis, tumor heterogeneity, and therapeutic targets. Throughout this session, we will explore these topics in detail, uncovering the intricate relationship between DNA methylation and cancer. Now, let's begin our journey by diving into the first topic of discussion. Professor. One of the earliest observations in cancer research was genomic instability, where cancer cells displayed numerous alterations and changes in their DNA. Over time, it became evident that DNA hypomethylation, the reduction in methylation levels, played a critical role in inducing genome instability in cancer. Hypomethylation leads to the activation of repeat elements and the loss of imprinting, ultimately contributing to genomic instability. Professor. Cancer metastasis, the spread of cancer cells from the primary tumor to distant organs, is a complex process. Recent studies have highlighted the involvement of DNA methylation in regulating genes associated with metastasis. DNA methylation can activate or silence critical genes involved in cell migration, invasion, angiogenesis, and epithelial mesenchymal transition, EMT, all of which are key processes in cancer metastasis. Professor. Epigenome plasticity, the dynamic changes in DNA methylation patterns, is a hallmark of cancer. Alterations in DNA methylation can occur at different stages of tumorigenesis and contribute to the acquisition of cancer-specific phenotypes. Understanding the dynamics of DNA methylation changes holds significant promise in identifying potential biomarkers and therapeutic targets for cancer treatment. Professor. Tumor heterogeneity, the presence of diverse cell populations within a tumor, poses significant challenges in cancer diagnosis and treatment. DNA methylation patterns can vary across different regions within a tumor and between tumors of the same type. By studying the DNA methylation landscape, researchers can gain insights into the clonal evolution of tumors and potentially identify new therapeutic strategies targeting specific epigenetic alterations. Professor. Epithelial mesenchymal transition, EMT, is a critical process involved in cancer progression, invasion, and metastasis. DNA methylation alterations can modulate genes associated with EMT, leading to the acquisition of mesenchymal-like features in cancer cells. Understanding the epigenetic regulation of EMT-related genes is crucial for developing targeted therapies to prevent metastasis. Professor. DNA methylation plays a pivotal role in regulating gene expression. Aberrant DNA methylation patterns can lead to gene silencing or activation, consequently impacting cellular processes relevant to cancer development. We will explore the mechanisms by which DNA methylation alters gene expression and the implications for cancer phenotypes. Professor. Immunotherapy has emerged as a promising approach for cancer treatment. In recent years, researchers have found that DNA methylation alterations can impact the immune response against tumors. Exploiting these epigenetic modifications as therapeutic targets holds great potential for enhancing the effectiveness of cancer immunotherapy. Professor. Mutations in genes responsible for DNA methylation machinery can lead to dysregulated DNA methylation patterns in cancer cells. Moreover, DNA methylation changes can act synergistically with genetic mutations, further exacerbating oncogenic processes. Understanding the interplay between DNA methylation and genetic mutations will aid in deciphering the complexity of cancer development. Professor. Chromatin remodeling refers to changes in the physical structure of DNA and its associated proteins, impacting gene accessibility and transcriptional regulation. DNA methylation patterns can influence chromatin structure, leading to important changes in gene expression. Discovering the mechanisms underlying DNA methylation-mediated chromatin remodeling will unveil novel targets for cancer therapy. Professor. DNA methylation patterns can vary across different cancer types, highlighting the unique characteristics of each malignancy. Exploring the specific DNA methylation changes in various cancers can aid in cancer diagnosis, prognostication, and the development of subtype-specific therapeutic strategies. Professor. Telomeres, repetitive DNA sequences found at the ends of chromosomes, play a crucial role in preserving genomic stability. Aberrant DNA methylation can affect telomere length and, consequently, impact chromosomal integrity. Examining the relationship between DNA methylation and telomere length in cancer is essential for understanding the mechanisms underlying tumor progression. Professor. Maintaining genomic integrity is vital for the prevention of cancer development. DNA repair mechanisms play a key role in fixing DNA damage and maintaining genome stability. DNA methylation alterations can hamper proper DNA repair processes, leading to the accumulation of errors and genomic instability. Understanding the connection between DNA methylation and DNA repair dysfunction is crucial for developing targeted therapies. Professor, in conclusion, our study of DNA methylation in cancer has illuminated its diverse impact. From metastasis to tumor heterogeneity, we've identified therapeutic targets and unraveled genome instability mechanisms. This knowledge advances diagnostics, prognosis, and personalized therapies. Together, we aim for a cancer-free future. Thank you and see you next time.